Let me just say that there are several different kinds of evidence, all of which are, and I'm not arguing that this is conclusive, this is still a hypothesis, but I would argue that it is the preponderance of the evidence clearly now points to a lab leak. The evidence is of four different kinds. There is molecular evidence, that is to say the sequence of the genome of the virus suggests editing of a kind that we know was taking place at the Wuhan Institute of Virology. There is phylogenetic evidence, that is to say the group, the sarbecoviruses to which SARS-CoV-2 belongs are known not to have furin cleavage sites, and yet this furin cleavage site shows up phylogenetically out of place in SARS-CoV-2. There is epidemiological evidence. We have a virus that did not show any evidence of evolution early on. It arrived in human beings perfectly adapted to spread at a high rate, whereas we would have expected a period of evolutionary adaptation that we cannot find. And then fourth, and maybe most important, there's a tremendous amount of circumstantial evidence surrounding the way the Wuhan Institute of Virology has dealt with the question of a possible lab leak. Initially, the laboratory in question acknowledged that a lab leak was possible. In fact, the leader of that lab, Zhizheng Li, said she didn't sleep a wink for some time, thinking that maybe the virus had escaped from her lab. So we know that such a thing is plausible. We also have the case of them having renamed a sample in a way that contravenes scientific protocol. So RATG13, which is the closest relative of SARS-CoV-2, turns out to have had a prior name in that laboratory, 4991. There's no reason to have renamed it. It effectively suggests keeping of two sets of books. So in any case, all four types of evidence point towards a laboratory leak that is now being obscured.
Right.
So I fully admit I am wildly out of my depth on this topic, but I just want to offer, so one of the pieces of evidence that has been offered against the idea of a lab leak. This is from Science News, and it's based on a report that was published in Nature Medicine. They say that this virus has components that differ from those of previously known viruses, so they had to come from an unknown virus or viruses in nature. And this is the quote from the study: "Genetic data irrefutably show that SARS-CoV-2 is not derived from any previously used virus backbone," meaning that, okay, if you're creating something in the lab, you're using an existing known virus and then you're modifying it. They're saying this is coming from nature because this isn't anything that we've ever dealt with before. What do you make of that piece?
Well, you know, at some level, we are now dealing with a kind of narrative theater surrounding this virus. That argument doesn't make any sense, and nobody who is in this community could imagine that it does, for the following reason. When we say that the virus may have been modified in the lab, we are not necessarily talking about edits where someone sits down to introduce a sequence. That is certainly possible. That is something that we do. But we are in general talking about a type of gain-of-function research that uses a process called serial passaging, in which we do things in the lab that we literally do not know how to do. And the way we do it is we use evolution. We take a virus and we can introduce it into tissues or into animals, and then we can allow it to transmit between cells or between individuals of these animal species. And so evolution does the work for us. And for anyone to argue that because, you know, as the initial Nature paper that dismissed this possibility argued, although SARS-CoV-2 is very good at transmitting between people, the sequence is one humans would not have predicted would be successful. That was the argument against the possibility of this being human modified. But it elides the strong possibility that serial passaging discovered things about transmissibility that we humans simply did not know until it had occurred.